Gotten as far as the rudiments of glomerular filtration. We talked about the idea of the renal corpuscle and how the glomerulus sat encapsulated in Bowman's capsule. And I'll show you a slide here to start that today. But what we want to finish there is we want to finish this idea of flow, because we want to generate a differential in pressure. A differential in pressure between the fluid in the glomerular capillary and the fluid in Bowman's capsule. And we'll see that once we generate a pressure differential, we're now going to get a movement of fluid from the blood vascular compartment into the renal compartment. And we're going to talk about this in terms of a glomerular filtration rate, which we're going to talk about as a GFR, being in the neighborhood of about 125 mils per minute. Then what we're going to do is we're going to explain all of these ideas in terms of an individual nephron. But one of the things you have to recognize as we're walking through this is that each kidney contains about 500,000 nephrons, and they're all working simultaneously. Two kidneys together have about 1 million nephrons in them. And we always tell the story down at the cellular level from the point of view of an individual nephron. But recognize that this is going on in parallel all of the time with many, many nephrons at the same time. We're going to lay out the idea of the hydrostatic and osmotic pressures giving rise to a pressure differential and a glomerular filtration rate. But we said yesterday that one of the hallmarks of the renal system is we push out an enormous amount of stuff from the blood vascular compartment into the renal compartment, and then we start picking and choosing that which we want to keep and that which we're going to pass to the outside world. And so we'll begin that discussion here in something called the proximal convoluted tubule. The epithelial cells of the proximal convoluted tubule will extend right into Bowman's capsule. There'll be a continuous layer of epithelial cells. Then we're going to find that in the proximal convoluted tubule is where we begin to do this process of reabsorption. In the proximal convoluted tubule, we're going to reabsorb about 60% of the water and sodium that we pushed out from the blood vascular compartment. And then we're going to reabsorb 100% of the glucose and the amino acids. Again, emphasizing the idea that any molecule with a molecular weight less than 1,000 will move from the blood vascular compartment into the renal compartment through those gaps between the endothelial cells and through the gaps between the podocytes in the Bowman's capsule. So now we've moved these molecules from one compartment to the other, and we begin reabsorbing them, and we're going to talk about how this reabsorption takes place. There are two major pathways for these ions and molecules to move. Much of the water and sodium is going to move in a kind of paracellular route. It's actually going to move between the epithelial cells that are forming the proximal convoluted tubule. We're going to tell the story here that's going to violate everything I've told you so far about tight junctions. We've talked about tight junctions being this great and wonderful seal. Well, we're going to see that's not a perfect seal. And we can think about them as forming a kind of a labyrinthine pathway between the lumen of the proximal convoluted tubule and out into the extracellular space, and then eventually back into the peritubular capillary. Then we're going to find that the cells here in the proximal convoluted tubule will allow most of the water and solvents to go around them, paracellular. But there will be a substantial amount of sodium which is going to come in with glucose and amino acids, because we're going to find there are transmembrane proteins located in the apical membrane, which are going to be responsible for snagging an ion of sodium and simultaneously snagging a molecule of glucose or amino acids and transporting those molecules across the apical plasma membrane. Once those molecules are inside the epithelial cell, then they're going to be transported across the basolateral membrane through something called facilitated diffusion. These are great big channels. And so now we will have moved the glucose and amino acids and a sizable portion of the sodium through this transcellular route. And then once they've exited either going around the cells or through the cells, then there'll be a return of these molecules by diffusion back into the peritubular, should be PERI, into the peritubular capillary. The peritubular capillary is now going to be a capillary that's an extension of the efferent arteriole. So remember yesterday, we had the blood flow into the glomerulus through the afferent arteriole. It was drained by the efferent arteriole. We're going to extend that efferent arteriole today to include into the peritubular capillary, and we'll see that much of what has been forced into the renal compartment is going to return to the blood vascular compartment here in the peritubular capillary. But you may remember, we introduced this yesterday, the position, the location of something called the nephron. And the definition of the nephron is the structural and functional unit of the kidney. Now that may sound familiar to you because we talked about the neuron in the central nervous system as being the structural and functional unit of the nervous system. Well, here it's a nephron. But one of the distinctions here is where the neuron is an individual cell, the nephron is a multicellular structure. It is a fairly large structure. It's going to extend all the way across the kidney as we drew it yesterday and back up from the medulla into the cortex. And we'll draw this again today. But when we talk about the nephron, we see that it is composed of various parts. And as we move through these parts, we're going to see that each of the epithelial cells has a slightly different complement of proteins in it that are slightly differently regulating the movement of ions and small molecules. We'll talk about Bowman's capsule today. We'll talk about the proximal convoluted tubule, and we need to get to the point where we lay in the portion of the extension, actually, of the proximal convoluted tubule into the descending limb of the loop of the nephron. It's going to be a turn, a so-called hairpin turn. It's going to make a U-turn into the ascending limb of the loop of the nephron. We'll then return back up to the cortex, and that's going to be in the distal convoluted tubule. That's the nephron. Now, if there's time, we'll extend this nephron into the collecting duct. But I usually try not to include the collecting duct in this description because the collecting duct is not officially part of the nephron. It's a separate structure. But the long and the short of this, one of the things I want you to be able to envision is we go from Bowman's capsule and the proximal convoluted tubule, where this is cortex, to the descending and ascending limb, which is medulla, to the distal convoluted tubule, which is cortex. Much as we drew it yesterday. And we'll walk through that again. But let's start here with a picture of a diagram, rather, of a renal corpuscle. I think it's worth looking at this because yesterday I drew it in a highly simplified and schematic way, where the tuft of capillaries known as the glomerulus was represented as a kind of oxbow in a river. Well, this is a more accurate demonstration. And the components here are the afferent arteriole feeding blood into this capillary. But it's not a simple capillary. It's a sort of a convoluted set of tubes. But these are all capillaries. They're composed of a single layer of endothelial cells. And if we follow the blood flow through this, the blood flow comes from the afferent arteriole into the capillary and then back out the efferent arteriole. And then this efferent arteriole is going to connect up to the peritubular capillary. But where we left off yesterday was describing the various pressures within the glomerulus and within this sort of cobblestone structure, the Bowman's capsule. So what we need to envision here is the movement of fluid from the capillary out into this onion-like structure, which is basically a bowl or chalice. And we're going to move the fluid from these tubes out into the bowl and chalice. Bowl or chalice. The fluid now is going to move from this compartment to this compartment, and the driving force of the movement of fluid from the blood vascular compartment into the Bowman's capsule is going to be predominantly the hydrostatic pressure. Remember, we said that if we'd follow this afferent arteriole back to its origins, it would go back to the renal artery, back to the descending aorta, back to the left side of the heart. So there's a considerable pressure that is the hydrostatic pressure forced against this wall. But we're also going to see that once we force the fluid out, we leave protein behind, and that's going to generate an osmotic force, bringing the fluid back in. There's also going to be a hydrostatic pressure here in Bowman's capsule, but in the end, there's going to be a net pressure of moving fluid from the blood vascular compartment, i.e. the glomerulus, out into Bowman's capsule and draining that fluid now into the next structure, which will be the proximal convoluted tubule. So as we walk through this, and I oversimplify the structure of the capillary and the structure of Bowman's capsule, see if you can hold in your mind's eye that description of the Bowman's capsule and the glomerulus, which make up the renal corpuscle. So let's do that again here. Let me try again. So what I'm going to draw here first is that cup-like structure. And I'll draw it just as a series of lines, but recognize that as we draw this, try again. As we draw this, this is meant to be a series of epithelial cells. So we have a continuous lining of epithelial cells. And these we can label as the epithelial cells of Bowman's capsule. Then feeding into this structure are the blood vessels. Where this structure is intended to represent the afferent arteriole. And this structure, the oxbow, let me make this a little bigger. The oxbow is intended to represent the glomerulus. So this is the glomerular capillary. And this structure up here is meant to represent the efferent arteriole. So blood flow through this thing is in this direction. And again, if we were to walk from the afferent arteriole back to its origins in the various arterioles and into the renal artery and back to the descending aorta, we would be on the left side of the heart. This is the oxygenated blood. And so that means that we have a considerable pressure against these endothelial cells that are forming the lining of the glomerular capillary. So the oxbow we're going to draw here as having a lining of endothelial cells. And that was represented in the slide that I showed by having this sort of tuft of capillaries, all of them lined with the endothelial cells. But from yesterday, you'll remember that there is a hydrostatic pressure on the endothelial cells. And that we said that those endothelial cells are not entirely like the endothelial cells we've seen before, where they're closely opposed to one another and knitted together by tight junctions. They have sizable gaps between them. These gaps are such, where if these are the endothelial cells, and we imagine these are in parallel so we can think about them as in 3 dimensions as being a cylinder, there's now this hydrostatic pressure against the wall of 55 millimeters of mercury. Now, the gaps between adjacent endothelial cells, these gaps, these permit molecules of molecular weight less than 1,000 to pass from the blood vascular compartment out. So anything less than the molecular weight of 1,000 is going to be forced out of this capillary into the extracellular space. But you'll remember from yesterday that right next door to these endothelial cells are these so-called podocytes. These are the podocytes of the Bowman's capsule. Now recognize all we have done here is take this sort of drawing where we show the structure of the glomerulus sitting in Bowman's capsule to now form the interface between the blood vascular compartment and the renal compartment. This is just a smaller drawing of what we see here. We've just blown this thing up so that we can imagine what's going to be moving from here out into the extracellular space and then into Bowman's capsule. You may recall that these endothelial cells sit on a basal lamina. And so now molecules, particularly water and sodium and chloride and glucose and amino acids. Any component of blood that has a molecular weight less than 1,000 is going to move out of this compartment into this compartment. But remember, one of the major components of blood is blood plasma. And we said that blood plasma was in the neighborhood of 8% protein. And typically, these proteins have a molecular weight of about 40,000. So they're 40 times too big to move through these narrow gaps separating the endothelial cells. So that means they're left behind. The way I kind of imagine this, it's as though you have a dish rag and you're wringing out the dish rag, and you're moving water and salts out, but you're leaving behind here a substantial number of these protein molecules. If you've squeezed the water out and left behind the proteins, what's the water differential? What's the concentration of water differential between these two sites? We have more water over here. We squeeze the water out from this compartment. We now have a gradient. We have a gradient such that we have more water here than here. So now, because of that gradient, we now have another force moving the water back into the capillary. This is said to be the osmotic. Sometimes you'll see this as oncotic pressure. We can measure this. We can say that this pressure is in the neighborhood of about 30 millimeters of mercury. Well, there's one other pressure we have to take into account here. And that is that because this Bowman's capsule is a confined space, we haven't shown it here, but it's pretty well sealed off against the blood vessel. And so it's a confined space with a narrow tube leading out, which we're going to call the proximal convoluted tubule. So you can imagine as we put fluid into this confined space, it's like filling a balloon with water. So as we fill this balloon with water, there's going to be a pressure that's generated. So we'll now have a new pressure, and I'll put this one in purple. This is a pressure back in this direction, which is the pressure of Bowman's capsule. This is actually a hydrostatic pressure. And we can measure it as being about, 15 millimeters of mercury. So let's look at the net pressures. There is a net pressure outward based on the hydrostatic pressure here. Let's not go up there. This is hydrostatic. And it's about 55 millimeters of mercury. But the other two pressures, the one in purple, so this is meant to be the hydrostatic pressure. This is the hydrostatic pressure of Bowman's capsule. Along with what I'll put in an orange here, the so-called osmotic pressure. Well, if we add all of these up, we have 55 hydrostatic, but we have the purple pressure. We have the purple pressure being 15, and the orange pressure, or the osmotic pressure, being 30. These are in the same direction, so you add these two together, you get 45 millimeters of mercury. So there's a pressure moving fluid from Bowman's capsule back into the capillary of 45 millimeters of mercury. But there's a hydrostatic pressure from the blood vascular compartment, from the heart, moving the fluid from the capillary into Bowman's capsule. If you take these together, we have a net, put it in blue, we have a net pressure, we have a net pressure of 10 millimeters of mercury. In other words, there is a delta P. There's a delta P between the capillary, Bowman's capsule, between the capillary here, the glomerular capillary, and Bowman's capsule. We now can talk about a delta P being about 10 millimeters of mercury. Whenever we see a pressure differential, you can assume there's going to be a flow. Unless there's an enormous resistance, there's going to be a flow. Well, if we work our way through these numbers, and I won't go through all the numbers, but it turns out, given this 10 millimeters of mercury pressure difference between the glomerular capillary and Bowman's capsule, and given the fact that you have about 1 million of these working all at the same time, we find that there's a net flow of about 125 milliliters of water per minute. So every minute, you're moving 125 milliliters of fluid from the blood vascular compartment in the glomeruli into Bowman's capsule. Well, do those numbers. Imagine sitting here for an hour, and you have 125 mils per minute times an hour, 60 minutes, that's 15, that's 7.5 liters of fluid you're moving through from the blood vascular compartment into the renal compartment every hour. Now, if you didn't reabsorb that water, you'd be pretty uncomfortable by the end of an hour's lecture. So there must be some mechanism in here whereby you can reabsorb this water. And that's where we're going to go now. We're going to start talking about how we can begin reabsorbing the water from the renal compartment back into the blood vascular compartment. And as we reabsorb the water, ions will follow. And that's where we're going to go now and talk about what happens in the proximal convoluted tubule. So any questions to there? Okay, so now then, let's go back up to the upper panel and extend these epithelial cells so that these are now the epithelial cells of the proximal convoluted tubule. So you can think of the proximal convoluted tubule as being a drain that comes out of Bowman's capsule, and we're draining the fluid out of Bowman's capsule into the proximal convoluted tubule. We've talked over here about how we have a net pressure, moving fluid from Bowman's capsule moving fluid from the capillary, the glomerular capillary, into Bowman's capsule. And how we're moving a lot of that fluid every minute of every day. Now what we want to think about is how we're going to begin to do this in the other panel. Take this panel and label this as reabsorption. In the proximal convoluted tubule. Let's enumerate over here what we're in fact going to be reabsorbing. In the proximal convoluted tubule, we reabsorb something in the neighborhood of about 60% of the water and about 60% of the sodium and chloride. But we will also reabsorb 100%, things are working properly, of glucose and 100% of amino acids. Just a reminder here that glucose is pretty important to us. That's the major carbon source we're going to use to generate ATP. We're going to break down glucose into pyruvate, feed that into the mitochondria, and in the end generate ATP. So we don't want to be peeing glucose to the outside world. We want to retain as much glucose as we can. So what we do is we reabsorb the glucose, which has a molecular weight way, way smaller than 1,000 and will easily move from the blood vascular compartment into the renal compartment. So we're going to reabsorb all of that. Similarly with amino acids, we don't want to be excreting amino acids either. They're important building blocks for proteins. So we want to reabsorb the amino acids. So what we're going to do now is think about what's going on over here in the proximal convoluted tubule. And I'll lay this out much as we have on the right side for Bowman's capsule. But now I want to imagine here that we have this series of epithelial cells. These are intended to be the epithelial cells of the proximal convoluted tubule. And so we will have squeezed out from the blood water and sodium and glucose and amino acids. Now over here, I want to take a subset, a smaller set of these epithelial cells and blow them up even farther so we can look at what's going on in the neighborhood of these individual epithelial cells. I'm going to draw them as these sort of cross-like structures. And I'm doing this so that I can bring the lateral sides of these epithelial cells close together. So this is intended here to be the basilar surface. So this is basilar. Just as this is basilar up here and basilar down here. So we're trying to show here these two parallel rows to exemplify a proper cylinder. All I've done here is taken the upper row and blown it up a little bit bigger to show these individual cells. So if this is basilar, that means this is apical. So that means this is the apical surface. So in here, this is cytoplasm. it's true that the lateral portions of these epithelial cells contain tight junctions. In the past, when we've talked about tight junctions, we have talked about them as though they were absolute, that they formed a watertight or ion-tight seal and nothing could move through them. That's not really quite true. And we now think, particularly here in the proximal convoluted tubule, we think of these tight junctions as zipping together the two membranes, but allowing for a fairly labyrinthine pathway from this luminal side to the extracellular side. And one of the things that these cells will do is down here in this region, put this in red, there are going to be pumps. And they'll actually pump sodium and allowing chloride to follow. We'll end up with a fairly high sodium chloride concentration down here in this narrow gap, this very small volume. By elevating the local sodium chloride concentration here, we can now generate an osmotic gradient. So we'll put a lot of sodium and chloride in here. We have water in here. And that now will provide a gradient for water to diffuse through the labyrinthine pathway and down here into the extracellular space. As the water moves from the luminal side around these cells through the so-called paracellular pathway, as the water moves through the paracellular pathway, sodium ions and chloride ions join. This is basically in a process known as solvent drag. So the sodium and chloride ions are in solution in the water, and so as the water is dragged across, because we've generated this high sodium chloride concentration here locally, we now pull water through the system. So most of the water, most of the 60% of the water and the 60% of the sodium and chloride that are moving from the luminal side out here into the extracellular space, most of that movement is through this paracellular pathway. The fact of the matter is there's so much fluid moving from the blood vascular compartment into the renal compartment, we just can't accommodate it by moving through the cells. We would basically dilute the ion concentration in the cytoplasm if we moved all of that water through the cells. So we move the water around. That's the paracellular route. Having said that, not all of the sodium, and particularly not any of the glucose or amino acids, move in this paracellular route. We're going to see the glucose and amino acids are going to move in a so-called transcellular route. So any questions up to here? So we've established this idea of a pressure. We've established the idea of a flow. We've established the idea that now we want to reclaim or reabsorb these molecules. Something like 60% of the water and the sodium is going to be reabsorbed in this region of the proximal convoluted tubule. Most of that reabsorption is coming through the paracellular route. But if we look at these epithelial cells out here on their apical surface, remember this is the luminal side, so we're talking about the surface here. These cells will have transmembrane proteins here called sodium-driven co-transporters. Now, that's a long word, and we're going to run into it a number of times, but if you dissect the word, if you unpack the word, it's a fairly simple meaning. Now remember that out here, we're going to have lots of sodium ions. We're effectively going to have probably as many sodium ions here, almost as what we had in the blood coming into the glomerulus through the afferent arteriole. So these are going to be, the concentration of sodium out here is probably in the neighborhood of 130 or so millimolar sodium. So we have lots of sodium. We also have molecules of glucose and molecules of amino acids. Among these epithelial cells in the proximal convoluted tubule, on their apical surfaces, as transmembrane proteins, are these sodium-driven cotransporters. And what we mean by sodium-driven is we mean that we have 130 millimolar sodium out here. Inside the cell, we have about 10 millimolar sodium. So we have a pretty good gradient. So that means we have plenty of sodium out here to snag the sodium ion. Meanwhile, this transporter can also, once it's bound to the sodium, it's now able to bind a molecule of glucose. Once this transporter has simultaneously bound an ion of sodium and a molecule of glucose, it effectively turns inside out. And when it turns inside out, the sodium moves from the extracellular region into the cell. But along with that sodium goes glucose. We'll recognize what we're trying to do here. We don't want to get rid of glucose. It's much too important a molecule to us. So we need some way to move the glucose from the lumen of the tube here, the proximal convoluted tubule, into the extracellular space, back into the structure I'm going to draw here as the peritubular capillary. Let's follow this backwards. So if we go back here to our most low resolution, farthest drawn out, most zoomed out image, where we have the afferent arteriole, the glomerulus, the efferent arteriole, we find that this efferent arteriole then extends into this structure here, which begins to wrap around, sort of snakes around the proximal convoluted tubule. And in the process of snaking around the proximal convoluted tubule, this structure now becomes the peritubular capillary. The peritubular capillary is lined with an endothelium. It's like all blood vascular compartments. So there's an endothelium here, and what I'm drawing in here with these small green ovals, that is meant to be the same thing as these green ovals. So this is the peritubular capillary. If we go to the larger view, we now have these endothelial cells, again forming, this is an endothelial cell, of the peritubular capillary. So now if you look at this, you begin to recognize that we are using the sodium-driven cotransporter to move glucose into this cell. But now if we imagine this cell as a kind of bucket, you'll begin to see that we're going to fill this bucket with glucose. And we're eventually going to make a high concentration of glucose inside the cell. So there needs to be some way of draining the glucose out of the cell. Well, it turns out that down here, on the basilar surface, is a transmembrane protein, which is a facilitated FASCIL, a facilitated glucose transporter. Now, the facilitated glucose transporter is just a great big pore. It's selective for glucose, but it's large enough that it will allow glucose through. So let me redraw this here. So we have one of these cells. Orienting you again here where this is basilar. Out here is the sodium-driven cotransporter. And now on this side in black, there's this transmembrane protein. You can think of it almost as a giant hole in the membrane. And this is a glucose permeable protein. So then the glucose now is free to diffuse from the cytoplasm out into the extracellular space. So now we're moving the glucose. Glucose is moved through the epithelial cell It's moved through the epithelial cell cytoplasm. This is said to be transcellular transport. So the glucose now is being actively moved in down a concentration gradient using sodium. But then, once it's inside this cell, it can now diffuse out onto the basolateral side. So that in this image, glucose is moving across the cell into the extracellular environment. But remember what's up here. This is blood. And now, remember, we've squeezed out most of the glucose from the blood vascular compartment into Bowman's capsule. That was done back in the renal corpuscle. So we've squeezed out most of that glucose. So this is relatively low concentration of glucose. So this now allows for us to permit the glucose to diffuse back into the blood. So we effectively have replaced, we have reabsorbed the glucose that we squeezed out from the blood into Bowman's capsule in the peritubular capillary. We can pretty much do the same thing, and let me put this in as a different color protein, because there are a series of other related proteins. And this is intended to be an amino acid transporter. The amino acid transporter does the same thing as the glucose transporter. It binds an ion of sodium. It binds an ion of sodium in the outside. And it binds an amino acid. Flips them then into the cytoplasm. And puts the sodium into the cytoplasm. But then again, here in the basolateral surface, this is going to be an amino acid facilitated transporter. So now what we've done is we've moved the amino acid in this transcellular fashion out into the extracellular space so that the amino acids too could be taken up back across the endothelium and back into the blood. So we use these very similar transporters, one dedicated to glucose and one dedicated to amino acids for this transport system. Now, I should point out here that almost all, in fact, all of the glucose and amino acid transport occurs here in the proximal convoluted tubule. Let me draw this in. Let me put this in the backboard. I'm going to draw a typical kidney, and we began this yesterday. And in purple, we laid out the corticomedullary gradient. I'm sorry, we laid out the idea of the cortex and the medulla. We said this is cortex. And we said that this is medulla. And we put into context here one of those nephrons. Now, I'm going to draw this again, but recognize as I do this, there will be literally hundreds of thousands of these. And I'm going to name the parts here. Where this is the Bowmans capsule. This is the proximal convoluted tubule. The proximal convoluted tubule then empties into this structure, known as the descending limb of the loop of the nephron. So it goes all the way down across the medulla. Then down here is the hairpin turn. And it then turns around and goes all the way back up to the cortex. This region is known as the ascending limb of the loop of the nephron. And this now empties into a structure, again, out in the cortex, called the distal convoluted tubule. Now, these are all parts of a particular nephron known as the juxtamedullary nephron. The juxtamedullary nephron composes only about 15% of all the nephrons, but we tell the story from the point of view of the juxtamedullary nephron because what we're going to find next time is that it sets up the corticomedullary gradient. Before I let you go, let me write this out. Up here in the cortex, we have a concentration of ions of about 300 milliosmoles. This is pretty much the same thing as plasma. But by the time we get down here, deep in the medulla, we can have concentrations as high as 1,200 milliosmoles. This gives rise to a gradient that goes from 300 to 1,200. This is said to be the corticomedullary gradient. And next time we'll talk about how we set up and how we maintain that corticomedullary gradient and why it's important to us in maintaining and generating hyperosmotic urine.